It's my great pleasure to introduce our first speaker, Pia Mach, who is currently a PhD student in molecular biology in FMI, Basel, Switzerland, in Giorgetti lab. She obtained a bachelor degree as well as master degree in biochemistry in Germany, studying arginine methylations in human cells before starting her PhD program. So she actually won the early stage researcher in the Chrome Design Network Fellowship for her PhD study. And she has also mentored multiple undergraduate students during her PhD research, as well as presenting her thesis work in multiple conferences. And I wanted to highlight her PhD thesis. It's working on Cohesin as well as CTCF control the dynamics of chromosome folding, which is the research with us today. And with that, I would like to hand the floor to Pia, and welcome. Thank you. You can start sharing your presenting your slides.
Thank you so much for the presentation and the introduction, Sky. Excuse me. Yeah, I'm very happy to have this opportunity to share my PhD project with you. I will talk a little bit about what Sky already mentioned, how Cohesin and CTCF control the dynamics of chromosome folding. And the reason we were interested in this in our lab in general, but then also particularly for my PhD, is that, as you probably all know, in mammalian cells, transcriptional regulation is often not only controlled by promoter sequences, but also by distant regulatory elements, namely enhancers that can be located very far away from their target promoter genes and can often also be basically interrupted or have intervening elements in between them, such as other enhancers and other promoters. So it's a very active question in the field that isn't currently very well understood, how an enhancer can find their target promoter and then regulate transcription from it. And one very prominent model where basically the chromosome folding dynamics come into play is that there is some kind of a looping event where physical proximity between the enhancer and the promoter gets established, which will then in fact turn into some kind of an induction of transcription and to promoter. And the reason why this is thought to happen is basically that it's been found previously by doing high-C studies. So basically a study where you can capture the conformation of chromosomes in interface and have a look on how often a particular locus in the linear sequence of the genome interacts with any other locus in the genome. And you get these 2D heat maps that are shown here. And what they showed is basically that there's domains of preferential interactions that are called these topologically associating domains, or short TADs. And what has also been shown previously is basically that these TADs, where sequences within these domains preferentially interact with sequences in the same domain where they do not interact with sequences in other domains, that they can be basically enhancer and promoter sequences that are co-regulated. So enhancer promoter pairs that regulate the transcription of the promoter. So this then basically led to kind of the question how these TADs are formed and whether this would influence how enhancer and promoter regulation or how enhancers regulate promoters. And when you ask the question how these TADs are formed, one very interesting fact that was found very early on was the fact that the boundaries of these TADs do coincide with sites that are bound by the DNA binding protein CTCF that binds in a convergent orientation at TAD boundaries, which then led to the a very prominent model in the field on how actually chromosomes are folded during interphase, which I will briefly summarize here. It's namely the loop extrusion model. In the loop extrusion model, there is a loop extruding factor, namely cohesin, which will bind to DNA in interface and will start extruding loops. until it encounters basically the N-terminus of a CTCF bound site. And it will then stall. And this will basically then keep on extruding until also a second CTCF bound site will be encountered in a convergent orientation, but not in a divergent or in the same orientation. And then this loop will be enforced between these CTCF bound sites until one of the two factors dissociates. And actually, this loop extrusion model can explain lots of the findings and these preferential interactions that we see in high C maps. For example, these corner dots that are thought to be CTCF mediated and cohesion mediated loops. And then also TATs, which are considered to be actually nested configuration of CTCF mediated loops. However, what we need to keep in mind here is that this is a snapshot in time of basically cross-linked cells. And it's also an ensemble average of many cells. And actually, what has been shown by sequential DNA FISH methods is that if you have a look at these conformations in single cells, then you can get very different pictures in different cells, as shown here on the left-hand side. However, if you again sum them up, you see what we see basically in high C maps, namely the ensemble average picture. Which then, of course, leads to the question, how dynamic are these structures actually in living cells? For example, do they get established just once per cell cycle, or do they form very often? And how does this actually relate to the loop extrusion process? And one other interesting observation that we can also help us understand this process a bit better is actually from the lab of Pia Mach in Vienna, they could show that indeed in vitro, Cohesin is able to extrude loops. And they've basically looked at this by placing DNA in a buffer flow and then flowing in all the components that are thought to be necessary to perform loop extrusion of DNA. And they could find that if you add Cohesin and ATP as well as a cofactor NPL, that you would be able to basically form these loops. So while this reassures us that loop extrusion could actually be the process in living cells that helps bring two specific sequences together, and it also gives us basically an upper handle on, for example, the speed of loop extrusion. What is still unclear, and this is where my PhD project came in, is what actually happens to chromosomal interactions in living cells. And for example, how often are cohesin-mediated interactions established? And then also, how long do they last? And the way I wanted to address this is using a life cell imaging approach. And for this, I used mouse embryonic stem cells, where I made use of bacterial operator arrays, for example, the TEDO and the LACO, which are short DNA sequences that can be bound by specific binding proteins, such as the TED-R and the LAC-I. And if you integrate arrays of these operators into the genome and have it bind by a fusion protein made out of a fluorescent protein and that's specific for pressure protein, you can basically visualize this integrated array as a subdiffraction limited spot in a single cell. And you can follow it over time using fluorescence microscopy. And we basically integrated two of these rather short arrays of 5 and 8 kilobases in mouse embryonic stem cells at a distance of 150 kilobases. We chose this distance because in mouse embryonic stem cells, that roughly corresponds to the average loop size. And it also is the average distance between so far characterized enhancer chromotype pairs. And we integrated these arrays in a locus that previously in our lab we described for a different study to be a very neutral environment. By neutral environment, I mean that basically there's no expressed genes, no active enhancers. You have a very minimal internal structure. So, and you also have no lamine-associated domains and no heterochromatic marks. And this gives us basically a playground where we can try to address how CTCF and cohesin influence the interactions between two chromosomal loci without having confounding effects from other processes. So with this setup in mind, we try to perform the experiment in 3 different flavors. In a first flavor, we integrated also strong CTCF binding motifs in a convergent orientation next to the operator arrays, so that ideally we would have impermeable barriers to cohesin, so that we would enforce an ectopic loop between these convergent CTCF sites. And indeed, if you have a look at this capture HiC-map, you see that where the array is pointed, you indeed get a specific interaction between our 2 loci. If you then remove these CTCF sites using recombinases, you indeed lose this interaction, as shown here. And then we also, in a third flavor, made this cell line into a diagram system for Rad21, which is a subunit of the cohesin complex. So if you treat these cells with the DTEC13 compound, you will lose all Rad21 from the cells, which will basically disable the cohesin complex so that normal loop extrusion can be performed, and you will lose all cohesin-mediated interaction, including our previously established ectopic loop. With this setup in mind, we then, of course, went to try to find out what are the dynamics of the system. And for this, we went to perform highly inclined and laminated optical sheet microscopy, as shown here on the right. And please focus your attention on the movie, which is basically an example of how these 2 operator arrays behave over the course of 3 hours with basically a time interval of 30 seconds. And in green, you can appreciate the forward CTCF site, or the array that's close to the forward CTCF site, and in magenta, the one that's close to the reverse CTCF site. And then, of course, if we have a look at movies from our 3 different setups, what we will see here is that in the case where you do have CTCF sites next to the operator arrays will spend most of the time in a very close proximity, which is here indicated by the white color in the movie, which shows co-localization. If you then remove these CTCF sites, what you can appreciate already in the movie is that the distances between the operator arrays becomes more variable and also larger, often larger in time. And the same thing can also be appreciated if you remove Rad21 from the cells, meaning if you deplete all cohesin-mediated interactions. We then set out, of course, to quantify this. And for this, we use spot detection and tracking to then basically get to these distance trajectories over time. So what we plot here for the course of the movie, the distance between the forward and between the LUC operator and the TET operator array. And what we see in those plots is actually shown here in an example that we see what we see already by eye in the movie, namely that in the presence of CTCF sites and Rad21, the loci spend most of their time in a very close proximity. That is actually fairly close to our experimental uncertainty limit of 130 nanometers. And if you then remove the CTCF sites or you remove Rad21, these distances become more variable and also larger in time. But of course, now that we also have the temporal component of this data, we can ask questions, for example, about how long the 2 loci spend in close proximity in these different setups. And for this, we used a hidden Markov model to classify 2 distinct states from these distance trajectories based on their radial distance, namely basically one proximal state, then where the loci are very close to our experiment, at the distance very close to our experimental uncertainty limit, and then one distal state that encounters all other configurations where the loci aren't in close proximity. And if you then basically classify all the trajectories that we found using this hidden Markov model and quantify the duration and the frequency of this proximal state, you can appreciate it then in the presence of the CTCF sites. We find the duration of this proximal state of roughly 16 minutes, which then gets decreased to around 6 minutes in the absence of the CTCF sites, or about 21. And we also do see a twofold decrease in the frequency of these interactions upon removal of the CTCF sites, or about 21. So if you think about what this means for how dynamic loci are over the course of a cell cycle, which in muscle embryonic stem cells is roughly 18 hours, you can think that they are highly dynamic and actually get established quite often over the course of such a cell cycle, which is also basically the conclusion of this part of the project, where I hope I could show you that the distances between two loci that are separated by a distance of 150 kilobases in the genome are actually highly variable in time, and that basically these contacts assemble or disassemble frequently and are controlled indeed by Cohesin and CTCF. However, in one last slide, I also want to mention here we can only assess proximity and not actually functional contacts, because this is not what we have access to from the microscopy data. So basically, we don't necessarily know if our two operators are just close in space, or if there's actually a cohesin-mediated and CTCF enforced loop that is this underlying. So in order, I just wanted to mention this as the last slide, in order to basically distinguish these two features, a colleague of mine, Pia Mach, he used polymer models that try to model a chromosome on which loop extrusion is acting, to basically then take this ground truth annotation of his data and apply noise to it, and then match it to our experimental data to see if we could find out how much of our proximal state is actually, or what component of our proximal state is actually due to cohesin-mediated loops. And when he basically finds the best match between our experimental data and his simulations, we find that the underlying cohesin and CTCF-mediated loop state lasts on average 5 to 15 minutes and occurs 20 to 30% of the time. And actually, this is in good accordance with also recent study of the lab of Anders Hansen, where they imaged, or they visualized a similar loop that was indeed at the endogenous FMB2 locus, where there was one or like two CTCF sites, one forward and one reverse, and they were separated by a distance of 505 kilobytes. And here they see actually that these loops are less frequent and only occur 3 to 6% of the time, which, however, is in good accordance with the fact that they are separated by a larger distance and also potentially have weaker endogenous CTCF sites as compared to what we see here. And with this, I would already like to thank you for your attention. And I would like to thank my entire lab, especially Pia Mach and Suzanne, the FMI facilities and our collaborators, and of course the funding sources in our lab, especially the ITN that Sky told you about Chrome design.
Great. Thank you for the nice presentation. So if you have any questions, feel free to either type it in the Q&A box, or you can raise your hand. So we already get a question in the Q&A box asking, do you have plans on looking at the non-neutral chromatin? By the way, nice to see you again.
Sorry, from who is this?
Lisa, sorry, I'm bad with names. Yeah, it's in the Q&A box. Yeah.
So the question was, do you have any plans at looking at, what was that? Sorry, I didn't get.
Non-neutral chromatin.
So far, I have to say no, but it's an interesting question, I think. if you would place basically the setup in a different locus, that if you would see a different picture, for example, if you would place it in a very heterochromatic area. This is actually, I think, also very tricky to do, because it might be that you would first have to find a place where to do this that is basically the same, in the sense that it is a very empty environment, but then as well also heterochromatic, right? In the sense, so you would have to find places in the genome that have these specific features so that you would be able to compare them, or you would have to go for an approach where you basically look at many loci, which with the current efforts in CRISPR-Cas technology that this requires might be quite challenging to do.
Great. Yeah. So you got a lot of more questions in the Q&A box.
Okay.
Go over 2 more. So given the observed dynamics, how many phase 4 partners do you think a given CTCF site has? Is it 1, 2, or more?
Oh, that's a good question. I think people have reported multi-bag contacts, right? So for sure there is, at a given time, it could also be that you have these kind of nested loop configurations, which would ultimately then also mean that a specific CTCF site is in contact with more than one other interaction partner. Just because if you nest these loops, you will end up with some kind of a multi-way contact. How many, however, I cannot answer that question at this moment.
Yeah. Oh, thank you. So the third question, and I think this will be the last question. I recall there were several papers saying CTCF and the cohesin binding lasts up to a couple dozen of seconds. So does that mean chromatin loops can stay even without CTCF or cohesin binding? And I think Ben had some related, like wondering whether this dosage also change the percentile of the time that the loops occur. So.
Yeah, so actually also a very good question. I think what we have here when we classify proximity is, first of all, an overestimation of how long these states last for several reasons. First of all, when you classify these trajectories with HMM, you do not take into account a lot of temporal memory. So, and indeed, I think it could be that basically a temporal memory of previous states remain even after CTCF and cohesin have dissociated from a locus, meaning that they don't necessarily directly set out to larger distances after the cohesin-mediated loop state has been resolved. I think in the sense that this loop state seems to be lasting roughly 5 to 15 minutes in our case, this would work well with current estimates of residence times of cohesin. However, residence times of CTCF, I know, are more in the shorter range in that sense. And here I would have to say that also in the genome, very often, I think, or particularly in this case, in the setup, you have 3 very strong CTCF sites in a row. So you have the combinatorial effects of them always being bound by at least one CTCF molecule because you have 3 in a row. So I think that's why here we see a very strong CTCF loop.
Yeah, so you actually got more questions in the Q&A box. And I think for the time purpose, we will not go over them, but if you can type the answer, we would really appreciate it. Yeah.
Yes, of course. Thank you very much. And thank you for all the questions.